close them down a little bit. But for now, it's still pretty warm in here. Okay, I think I can start. So welcome back. We had a long break, more than a month, between ESA meetings and holiday, and actually one day that was just canceled seminar. So today, Dr. Tarikul Islam is going to talk to us. He's from Bangladesh, where he got his master's degree and his bachelor's degree at the Bangladesh Agricultural University. He then started working as a lecturer. Eventually, he got a position as assistant professor. And they have this nice benefit where he can take off 7 years during that time and work in other labs around the world. So he took 4 years to get his Ph.D. at Western Sydney University in Australia, where he worked on the effect of silicone on insects. And that's what he's going to talk about today. And then since spring this year, he's been working in my program as a postdoc. And he's also working on some projects to do with silicone, effect on some of our insect pests and turf grass, as well as we're starting some looking at the interaction with some natural enemies, in particular entomopathogenic nematodes. But he's not going to talk about that today since we're just starting that. So without further ado, Tarikul, the stage is yours.
Thank you, Albert, for the introduction. So, very good morning to everyone. My name is Tarikul Islam, and as Albert mentioned, I gained my PhD from Western Sydney University in Australia, and I studied the effects of silicon fertilization on insect herbivores. So today I'll be talking about my PhD project, actually. So the title of my talk is A Multifaceted Defense, Impacts of Silicon Fertilization on Plant Defenses Against Herbivorous Insect Pests. So I'd like to start with some background information on plant silicon defense. So silicon is a beneficial nutrient for plants. They provides a range of benefits from enhanced growth to the alleviation of biotic and abiotic stresses. And silicon is naturally abundant in the soil, but they are mostly present as insoluble silicates and oxides, and these are not available for plant uptake. So plants can only take up silicon in the form of orthosilicic acid, which is further translocated to their shoot via xylem and finally deposited in and around tissues as hydrated silica gel or phytoliths. So in the picture you can see these flower-like structures. These are phytoliths. And plants actually vary in their ability to accumulate silicon. Plants are generally classified as accumulators, intermediate accumulators, and non-accumulators of silicon. So this is based on the amount of silicon they can deposit in tissues on a trioid basis. So if plants can take up more than 1.5% silicon of their shoot trioid, they are accumulators. So grasses are accumulators of silicon. So monocots accumulate much more silicon than dicot plants. So the primitive plants, non-vascular plants, they accumulate higher amount of silicon compared to vascular plants. And among flowering plants, monocots are more accumulators, and dicots are intermediate or non-accumulators of silicon. Alright, so silicon accumulation in plants is considered a physical defense because silicification makes tissues very abrasive and harder. So silicification also reduces the digestibility and palatability of tissues to insect herbivores. So when insects feed on silicified tissues, it can wear down their mandibles. So this is a photo from our recent paper in Functional Ecology, where we showed that the mandibular teeth of Helicoverpa parnigera was severely eroded when feeding on silicified plant tissues. So for insects, it's like getting a bunch of sand particles in their teeth. So previous studies have also reported that feeding on silicified tissues can reduce insect growth rates, can damage their gut, reduce their survival, and also they can increase their developmental time. And recent evidence also suggests that silicon can influence plant chemical defenses, particularly via the jasmonic acid signaling pathway. So my PhD project actually revolved around two central questions. One was how does silicon impact plant defenses across different silicon accumulating plant species against herbivores from different feeding guilds? As I mentioned, that plants vary in their ability to accumulate silicon. So there are accumulators, intermediate, and non-accumulators. And I used herbivores from different feeding guilds like chewing, fluid feeding, and cell content feeding. So previous research on silicon actually focused mostly on chewing insects and silicon accumulating plant species, for example, grasses. So we tried to use different study system, like incorporating different silicon accumulating species and different feeding guilds of herbivores. And another question we asked was, does feeding on silicified plants weaken insect anti-predator defenses? So insect herbivores, they feed less on silicification, like silicified tissues, and they also assimilate less nutrients from those ingested plant materials. So this can impact their anti-predator defenses. Alright, so the first study I conducted, there was a glasshouse experiment where we looked at the magnitude and locality of silicon induction by insect herbivory. So there are reports that mechanical damage or insect feeding can induce increased silicon accumulation in plants, because when plants are under threat, they accumulate more silicon to defend themselves against herbivores. So we asked, does silicon induction occur locally, that is only in the attack tissues, or it also occurs elsewhere in the plants? So is it a local or systemic induction process? And we also wondered if varietal differences affect silicon induction. So we used cucumber, which is an intermediate silicon accumulator, and two cultivars, Bartless F1 and Bait Alpha, and we grew plants hydroponically. So hydroponic experiments actually allow to exclude silicon completely, because there are always some silicon in the soil. So that's why we opted for hydroponic experiments. And the insect species was Helicoverpa parnigera, which is a chewing insect. It's a polyphagous pest of hundreds of cultivated and wild plants. So I'll be showing you some results from this study. So we found that silicon supplementation actually reduced both consumption and growth rate of Helicoverpa parnigera. So you can see that the growth rate was, so these are silicon plants. The growth was almost half on plants that have access to silicon supply, and it also reduced their consumption of leaf tissues. And the effect was consistent in both varieties. So the silicon induction, so we measured the concentration of silicon in leaves. So first, the local induction, you can see the insect feeding actually increased the accumulation of silicon in leaf tissues. So in both varieties, so Barclays F1 actually accumulated more silicon compared to beta alpha, but the induction level was proportionately similar if you compare with the non-insect plants. So it actually increased following insect feeding. So this is the local accumulation. So this is the silicon concentration in the attacked leaves, and we also found that the concentration of silicon was increased in non-attacked leaves. So that means that the silicon was actually induced systemically in plants. So in both varieties, actually, we found higher concentration of silicon following insect feeding. And here also, Barclays accumulated more silicon compared to beta alpha, but the induction level was proportionately almost similar. So that's our findings, that herbivory, insect herbivory induced both local and systemic silicon accumulation, and silicon induction was almost equivalent between plant varieties. In my second study, I looked at the impacts of silicon on plant indirect defenses. So all of you know that the plants release special volatiles upon herbivore attack, which are known as herbivore-induced plant volatiles. And these volatiles actually can attract natural enemies of herbivores, particularly parasitoids. And this is known as indirect defense. So we asked if silicon impacts herbivore-induced plant volatiles and natural enemy attraction. So for this study, we used French bean, which is a low silicon accumulator, and we used the two-spotted spider mite, Titanicus artici, which is a cell content feeder. You know that the spider mite, they actually feed on mesophyll cells. They suck out the cell sap, but it's a bit different from fluid feeders because their the length of their stylet is shorter than aphid, so they cannot penetrate into the phloem tissue. So they feed on mesophyll cells. And we also use a predatory mite, Phytocilus parsimilis, for testing natural enemy attraction. So this is how we grew our plants. So the plants were grown hydroponically, again, to exclude silicon. And this is an infested leaf. You can see spider mite damage, Titanicus artici damage on leaves. So the effects of silicon on Titanicus artici performance. So what we found is that silicon supplementation actually reduced Titanicus artici egg laying and population growth. So we measured egg laying and population growth at 2 time points, 6 days and 14 days after spider mite inoculation. And you can see at both time points, we actually found discernible effects of silicon on egg laying. So egg laying was reduced on silicon treated plants. And the population growth was also reduced at 14 days after spider mite inoculation. We also found that the physical damage on French bean leaflet was lower when they were supplied with silicon. You can see at 14 days after spider mite inoculation, we found that the damage on leaflet was lower on silicon plants. So spider mite, Titanicus artici, it also causes some sort of physiological injury to plants beyond physical damage, because they also inhibit plant gas exchange. So they reduces photosynthetic rate and transpiration. So we measured leaflet photosynthesis and stomatal conductance using a portable photosynthesis system, which is Lycor, you know that. So what we found is that silicon had no effect on leaflet gas exchange parameters when there was no spider mite herbivory. So you can see that the photosynthetic rate at 6 and 14 days after inoculation, photosynthetic rate and stomatal conductance were similar on both minus and plus silicon plants, but spider mite feeding actually reduced all these gas exchange parameters irrespective of the silicon treatment. But silicon actually did partially ameliorated the reduction in stomatal conductance and photosynthesis. So this is the photosynthetic rate on plus silicon leaflets. So you can see there was a partial amelioration when plants were supplied with silicon. So that's our findings, that silicon actually partially ameliorated leaflet gas exchange upon Titanicus artica herbivory. Alright, so we did olfactometer bioassays using the predatory mite, Phytocillus persimilis. So we used a Y-tube olfactometer. So we placed a Y-shaped metal wire inside that Y-tube olfactometer for this predatory mite to make a railroad for them. And this is how we did this. So the airflow was controlled using flow meter. So it's a typical olfactometer setting. So our results actually is interesting. So what we found that the predatory mite did not discriminate between plus and minus silicon plants when there was no spider mites on them. But the predatory mites were attracted more to minus silicon plants when those plants were infested by spider mites. But the plus silicon plants had no mites on it. So 70% of the predatory mites were attracted to minus silicon plants when they had spider mite infestation. But when both plants, both minus and plus silicon plants, had spider mites on them, the predatory mite was attracted more to plus silicon plants. So this is important because, like, if these predatory mites were attracted to plus silicon plants, even in the absence of spider mites, this can actually exacerbate their foraging efficiency. So we found that the plus silicon plants were attractive to the predatory mites only when they had spider mites on it. So we also trapped the herbivore-induced plant volatiles emitted from different treatment plants, and we analyzed those volatiles using a GCMS system. So we found 17 different volatile compounds from plus and minus silicon plants experiencing Titanicus artica herbivory, and 8 volatile compounds when plants had no Titanicus artica. So silicon application did not cause the synthesis of any novel volatiles. So silicon just changed the quantitative composition of the volatile blends. So there was like the volatiles compounds in bold, the emission of these compounds were elevated from plus silicon plants, and the compounds in red, these were actually decreased from plus silicon plants. So what we found is that plus silicon plants, they are actually emitting the same volatiles but in different ratios. So here is the separation of compounds, so the PC analysis. You can see that the spider mite feeding actually causing the more variation. So the first principal component was mostly explained by spider mite feeding. So these are mite infested plants, and these two are mite free plants. So the principal component analysis actually resulted in 4 principal components, and 70% of the variation you can see that were explained by first 2 principal components, and these are grouping of samples according to the first 2 principal components. Alright, in our third study, we looked at the impacts of silicon on interspecific interaction between 2 insect species. So the most of the previous research has reported the effects of silicon against chewing insects. I mentioned that earlier, and the effects of silicon for sap-feeding insect herbivore is not actually clear-cut, because some research reported positive effects of silicon, some reported negative effects, and some reported like neutral effects of silicon. So we asked how does silicon affect insect performance on contemporaneously shared host plants. So we used two different insect herbivores. One is cotton bollworm, Helicophora pardigera, and another is barred chariot aphid, Rhophalosiphum paddy. So one chewing insect and another fluid feeding insect. So we try to look at like how silicon affects them when they are sharing a host plant. And we used, as a host plant, we used purple false broom, Brachypodium distachium. So this is a grass. So it's a high silicon accumulator. And this grass is not that much important agriculturally, but they are used as a model for monocot plants, especially for wheat and barley, because they're phylogenetically they are very similar to Triticale crops. So that's why we use this one. So this is our glasshouse experiment. So we grew plants hydroponically. This is how we cased plants after insect placement. So what we found is that aphid abundance was actually higher on plus silicon plants compared to minus silicon plants, regardless of caterpillar feeding. So this is the aphid abundance on plants that had no caterpillars, and this is aphid abundance on plants that had caterpillars on them. So you can see that the abundance of both nymphs and adults and the total abundance of aphid was actually higher on plus silicon plants. So silicon supplementation actually favoring the colonization by aphids. We found the same trend when plants were also infested by caterpillars. So the abundance was lower here compared to no caterpillar plants. So there was a kind of competition between caterpillars and aphid. That's why you can see the number of aphids actually went down compared to this one. But silicon supplementation had a positive effect on aphid colonization. So although silicon had a positive effect on aphid abundance, we found that silicon reduced caterpillar relative growth rate, irrespective of separate on or contemporaneous feeding with aphids. So here you can see that the caterpillar RGR was actually lower on plus silicon plants compared to minus silicon plants, both in the presence or absence of aphids. So the aphid abundance and caterpillar RGR, like I mentioned, there was a negative correlation between aphid abundance and caterpillar RGR. And when we did multiple regression analysis, we actually found that the negative effect of silicon on caterpillar RGR was actually favoring aphid abundance. Because there is like interspecific competition for food and resources and for shelter. So when their niches overlap, plus silicon plants had low caterpillar RGR. That was actually beneficial for aphids because they are getting like more space and more food for their colonization. So we found that silicon supplementation had a positive effect on shoot biomass, and insect had negative effect. So insect herbivory actually reduced both shoot and root biomass. So that's obvious because insect feeding was like damaging plants. So the both root and shoot biomass of insect-fed plants were lower compared to insect-free plants. But silicon had a positive effect on plant biomass. Okay, so when we looked at the silicon induction in leaf tissues, we actually found that it was the caterpillar that was actually inducing more silicon accumulation in leaf tissues, not aphids. So you can see that the level of silicon induction was similar in caterpillar infested and in caterpillars plus aphid infested plants. So there was no difference in the level of silicon induction because the caterpillars were mostly responsible for the increased silicon accumulation in leaf tissues, not aphids. Alright, so our in our fourth study, we looked at the impacts of silicon on insect anti-predator defenses. So you all know that insect herbivores have evolved different anti-predator defenses, for example, morphological, behavioral, and immune defenses. So in this short video clip, you can see that this caterpillar is doing kind of thrashing and head rearing and biting this parasitoid. So this is a kind of behavioral anti-predator defense. So host plant quality actually can shape insect anti-predator defenses, because as I said, like insects feed less on silicified tissues, and they are assimilating less nutrients from those. So this can impact their anti-predator defenses, because they have less resource to invest on their growth and defense mechanisms. So yeah. So we asked, does feeding on siliceous plants compromise insect anti-predator defenses? And we used the same plant, Brachypodium distachyon, and cotton bollworm, Helicophora parmigera, for this study. So we grew plants hydroponically and then reared Helicophora parmigera larvae on those plants, plus and minus silicon plants. And then we tried to look at larval morphological defenses. So I tried to estimate the thickness of larval integument when feeding on plus and minus silicon plants, and the integument resistance to puncture. So parasitoids, they lay eggs inside this caterpillar, so they need to breach the integument layer for egg laying. So I try to estimate how much pressure is needed to puncture this integument when they fed on plus and minus silicon plants. So I used a drill press equipped with a hypodermic needle, and then I placed these caterpillars over a precision scale, and I looked at the scale reading when this needle actually breached their integument. So that's the integument resistance. And then I dissected out this larvae and estimated their integument thickness using a thickness gauge. So this is what I found, is that feeding on plus and minus silicon plants actually did not affect larval integument thickness, but the integument resistance was higher when larvae fed on minus silicon plants. So larvae fed on plus silicon plants had lower integument resistance despite no difference in integument thickness. So this is because the caterpillars feeding on minus silicon plants were bigger, and I found a positive correlation between larval final weight and their integument resistance. So this could be related. So I have also looked at larval behavioral defenses. So what I did is I pinched larvae using tweezers at their abdominal side, so at the posterior part of their abdomen, to imitate the attack of a predator. And then I video recorded those larval behaviors. And we found this 4 defensive behaviors. One was head rearing, which is the backward movement of their head, and thrash, which is a side-by-side swing movement, and regurgitation. And I also tried to estimate their escape behavior, how quickly they can escape. So I used a gridded plastic sheet and looked at how many squares they can cross within a minute. So that's their flee response. So I did not find any effects of silicon on larval behavioral defenses, so larvae were behaving very consistently. The only closer one was this one, number of head rears. Okay, then I looked at larval immune defenses. So we estimated hemocyte density in the hemolymph. So we found 5 different types of hemocytes: granulocytes, inocytoids, plasmatocytes, prohemocytes, and spirulocytes, but there was no effects of silicon on the hemocyte density. So density of hemocyte was unaffected by silicon, but we found a discernible effect of silicon on larval PO activity in the hemolymph. So phenol oxidase is a very important enzyme. Is an important enzyme for larval melanization response. So their immunity against pathogens depends on this enzyme activity. So what we found is that larvae feeding on plus silicon plants actually had higher PO activity in the hemolymph compared to larvae feeding on minus silicon plants. So which means the larvae feeding on plus silicon plants, they have kind of like higher immunity compared to minus silicon plants fed larvae. But when we looked at the actual melanization response, so we implanted a nylon filament inside the hemocoel of larvae and looked at the melanization of that implant. So we processed those images using image J, and we found that the melanization response was unaffected by silicon. So although they had higher PO activity, they were unable to melanize the implants, strongly compared to larvae that fed on minus silicon plants. So high PO activity actually did not translate to high melanization response. So this is the last study I did, my 5th study. So when I was doing this, the 4th study, I actually looked at the coloration of larvae when feeding on plus and minus silicon plants, and I found that like at certain point, at some point, these larvae, the Helicoverpa armigera, when they feed on plus silicon plants, they always show this black to brown coloration, and the larvae feeding on minus silicon plants, at some point they're becoming like green. They're showing green morph, green coloration. So this is basically kind of cryptic coloration. So they are hiding, they are blending into their leaf background. So this is also a like anti-predator defense. So in this experiment, we try to look at like why these larvae are turning green and why they are black or brown colored. So cryptic coloration in insect is an effective camouflage strategy, and this green coloration is actually caused by green hemolymph, which is termed as insectovardin. And this green hemolymph is a mixture of yellow and blue chromoproteins. And the prosthetic group of the yellow chromoprotein usually are lutein or beta carotene. So these are carotenoids. So this yellow component, this comes from carotenoids. And the blue chromoprotein, that comes from larval bile pigments, either pterobilins or mesobilivardins, like that. So this green hemolymph is a mixture of yellow and blue chromoproteins. And insects actually cannot synthesize their carotenoids. So they need to get those carotenoids from host plants. Aphids are exceptions because they got carotenoid biosynthesis genes from their fungal symbionts. But most other insects, including lepidopteran insects, they need to get these carotenoids from host plants for making green hemolymph. So we asked the question, does silicon hinder carotenoid acquisition in larvae? So we used the same study system, Bactropodium distachyon and cotton bollworm, Helicoverpa armigera. And we tried to measure the carotenoids and chlorophyll contents in leaves, in insect hemolymph, and insect frass to get an idea of what's going on there. So we found that, so first we measured the color contrast of larvae against their leaf backgrounds, and we found that the larvae actually contrasted more than threefold against a green leaf background when they were feeding on plus silicon plants compared to larvae feeding on minus silicon plants. So there's the larval color contrast. So when we measured carotenoid and chlorophyll pigments in leaves, we found that there were 4 different carotenoids in leaf tissues. So we found neoxanthin, violaxanthin, lutein, and beta carotene. So you can see that actually the both carotenoids and chlorophylls, for both we found the same trend. So we found that the minus silicon plants had constitutively higher levels of carotenoids and chlorophylls in tissues. So you can see that in the absence of insect attack, so just minus silicon and plus silicon, if you look at these two, you'll find that the carotenoid level was lower in plus silicon plants compared to minus silicon plants. But following insect attack, we actually found that there was no significant difference between plus and minus silicon plants. For both carotenoids and chlorophylls, we found that the concentration of carotenoids and chlorophylls was similar in leaf tissues following insect attack. But the constitutive level was higher in plus silicon plants. So we also measured carotenoids and chlorophylls in larval frass, and we found that all those 4 carotenoids were excreted in the frass. So larvae were partially assimilating carotenoids, and the large portion of the carotenoids were actually excreted in the frass. So we actually found that the larvae feeding on plus silicon plants had lower carotenoids and chlorophylls in the frass. So for all those carotenoids, you can see that the plus silicon larvae feeding on plus silicon plants actually excreted less carotenoids and less chlorophyll in their frass. Okay, the most important one, the pigment contents in larval hemolymph. So we found only 2 carotenoids in larval hemolymph. One is lutein and another is beta carotene. So you can see that the larvae feeding on minus silicon plants, they had very higher contents of lutein in their hemolymph. Although the beta carotene content was slightly lower, but if you look at the total carotenoid content, it's still higher in larvae feeding on minus silicon plants. And larvae feeding on artificial diet, we found no carotenoids in them. We also found a very small amount of chlorophyll A in some larvae feeding on minus silicon plants, but that was less than in like 20% larvae. So that's our finding, that larvae sequestered very lower levels of lutein in the hemolymph when they feed on plus silicon plants. So some take-home messages from my project. So we found that the silicon defenses could be effective in intermediate and low silicon accumulating plant species. Of course, it's effective in high silicon accumulating species. And we also found that herbivory can induce local and systemic silicon accumulation. We found that silicon may change herbivore-induced plant volatile emission, and also it can promote natural enemy attraction. So we used plants having different silicon accumulation ability, and we found that silicon defenses were actually effective in all plants. And we found that silicon is a very powerful defense against chewing and cell content feeding herbivores like Helicoverpa parmigera and spider mites, Tetanicus artici, but it was not effective against self-feeding insects like aphids, because it actually helped them to colonize in plants. And we also found that the feeding on siliceous plants could compromise insect anti-predator defenses. For example, their cryptic coloration, or their immunity, or their integument resistance that was lower. Yeah, with that I would like to thank my lab group. So that's my previous lab group, and I also thank my present lab group members. I would like to thank Western State University, where I did my thesis study. And you can look at some of my papers that I published during my PhD. So thank you so much for your attention, and I'll take any questions.
Yeah, so